Our next talk is by Dr. Stefano Ratti. And I'm really disappointed he couldn't be here in person. I was hopeful, but unfortunately for us, he is able to give a virtual talk for us today. Dr. Ratti is a full professor of human anatomy at the Department of Biomedical and Neuromotor Sciences at the University of Bologna. He is the principal investigator of the Cellular Signaling Laboratory. His research focuses are molecular mechanisms in rare neurodegenerative and oncological diseases, precision medicine innovations tested and developed on bodies donated to science, and innovative and creative medical training. Thank you so much for joining us, Dr. Ratti. We're looking forward to your talk.
Thank you, Margot, for the presentation, and thank you all. Yeah, we are very sorry you could not be there. Hello to everybody. Hi to Kazar and to the people I know that are present there. Unfortunately, we couldn't make it, but we are very grateful to Mayo Clinic, to Margot, and also to the ADLD Center to continue this, let's say, new tradition, but we hope will continue as a tradition of meeting to talk about ADLD. So I'll start with my talk about basically ADLD models and our experience at the University of Bologna. My presentation will be divided with a bit of background that I'm happy I'll be able to cut now after this beautiful the keynote and the first talk. Then will be a part about the engineered cell models. So the first work we did, most of it, apart one new part, has already been published. And the point 3 and 4, it's all not published data, and it's on human primary cells overexpressing lamin B1. And the last part is about iPS cells from ADLD patients creating organoids to study disease. And I'll conclude with some next steps and future direction regarding our studies. Before starting, I put here this comic done by an artist, a comic artist just a few days before the meeting. And I put this here just to underline the importance of collaboration and interdisciplinarity. There are 3 figures here, but really I think that everybody that is in the room or in our laboratories could be here and will be there. This is just a way to remember to all our community this, as Kaza reminded before, it's done by scientists, but also patients who try to contribute to, let's say, common cause together. We can skip, I think, the first part that is already been explained very well, and I'll just go to some important aspects. First of all, the importance of the increased level of lamin B1 protein, especially in the brain. It was even asked before, where is this overexpression? So that's a focus we want to remember. And also the differences between the canonical and non-canonical forms. So the deleted form, so the one without publication, has been highlighted. But it's very important to remember the lamin B1 is expressed ubiquitously, though that's why sometimes it's hard when we talk about blood of the patients or analysis to understand if really the double copy result with an overexpression of the protein everywhere, or if it is, for example, silenced in a specific area like the brain. And we will comment this later on. We all know, and I think that it's very clear that both phenotypes examined until now result with an lamin B1 expression on the level of the brain, resulting with a demyelination. And we all know that oligodendrocytes are responsible for the myelination around the neurons. But as it was even showed before, 2-3 years ago, we highlighted for the first time that astrocytes could be also involved. I think that's not something new, because a lot of us that study in neuroscience know that the role of astrocytes that in the past were considered to be the glue from this Greek word glia of the brain, so like only maintaining the structure of our brain, are actually involved in many activities. So that's where we started to investigate. And we started to investigate there because we thought that it could be a communication between different kinds of cells, astrocytes, oligodendrocytes, neurons, and microglia that could then result as a lack of myelination. There are indeed several factors like leukemia inhibition factor, one of the factors we studied that is produced by astrocytes in order to let evolve, mature the oligodendrocytes and produce the protein like MBP, PLP, MAG, that we know are involved in the process of creating this nice myelination. So we go to the first part here. You can see in the picture Dr. Usciano and Prof. Ramazzotti, who did with me the most part of this work that was published 2 years ago. And as always, we have always to start with something easy, also for a matter of funding that at the time we didn't have. So we started creating some engineered cell models overexpressing lamin B1 through interviral transfection, representing oligodendrocytic-like cell line and associated-like cell line. At the same time, we have fibroblasts of the patients, and we also overexpress in wild-type fibroblasts lamin B1 in order to create our reliable model. What is always important to remember is that the level of lamin B1 in the patient are not as high and as in most of the engineered models. First, because it's not easy to control always in all the models the level of lamin B1, and secondly, because we are facing a late onset disease. That means that if we want to see in vitro at least the effect or ex vivo, so from patients' cells modified, you have to somehow to stress the disease to see some results. And going back with the name, now I just use ADLD. The long name that was shown by Riley is not actually used by the Italian community, it's the ONIM name, so it's the updated one. But it's true that there are actually many publications with different names, and probably it could even change in the future. But that's something that concerns specifically, I think, the genetic counseling. So this was our first work. As you can see here, U87, so cells that represents in our case, astrocytes models were overexpressing a lot of lamin B1. That was already interesting data for us because oligodendrocytes could not resist to such a high level of lamin B1. Instead, astrocytes could result in a very, very high, not physiological level of lamin B1. So that was interesting for us because we had astrocyte cells overexpressing a lot of lamin B1. So we could be able to stress the system. So imagine what this could be in a long time situation like in a patient. And at the electron microscopy, this nice image we showed you, you can see severe alteration in astrocyte cell lines overexpressing lamin B1 compared not only to internal control and wild type, but also on oligodendrocytes. That seems, even when overexpressing lamin B1, to not be able somehow to arrive such a high level lamin as other cells, so they stopped before, meaning that the sum difference between these kind of cells could be really related to an alteration of their communication. It's hard to know which is the first cause, but we'll talk about that later. The first pathway we analyze is LEAF, as mentioned before, is a 6-type cytokine family and is involved in mTORC1 myelination pathways and has been studying also other disorders like MS and other kind of neurodegenerative disorders. And what was interesting for us that we saw that the cells overexpressing lamin B1 had a reduce in leaf production and also leaf receptors because they also have the receptors. And oligodendrocytes also overexpressing lamin B1 had a less level of the receptor. Also, the serenitins of our astrocytes-like overexpressing lamin B1 cells had a reduction of leaf, meaning that somehow leaf was less produced and less received by oligodendrocytes, less produced by astrocytes and less received by oligodendrocytes. As leaf is at the top of the chain of MEC-ARC, JAK-STAT3, and PI3 kinase and mTORC1 pathways, and that these are pathways. And I agree with Kazar that when we talk about pathways, it's never easy to understand where to find a site where you want to really attack the system, because these are very common pathways, but at least we're trying to elucidate what's going on here. And these are pathways involving many signaling survival pathways. What we saw is that leaf signaling was negatively affected in our model. So basically, as you can see here from the Western blot, the cells, both astrocytes and oligodendrocytes overexpressed laminin B1, had a downregulation of PA3 kinase signaling pathways. Also, a downregulation in STAT3 in both astrocytes and oligodendrocytes, and only in astrocytes, a downregulation of GSK3 phosphorylation. That was together with the different level of overexpression of laminin B1, together with the different morphology, together with the signaling now, where we started to think that there could be really a big difference between astrocytes and oligodendrocytes, even if we still were in a engineered cell model system. Instead, the third pathway analyzed was not affecting either oligodendrocytes or astrocyte cell line modified. Interestingly enough, there was another differences between the two cell population, is that in astrocytes leaf administration, we were able to partially rescue PA3 kinase and phosphorylate instead of GSK3 expression, but do not able to reactivate STAT3 pathways. There was instead possible to be reactivated in oligodendrocytes overexpressing laminin B1. So again, showing that the sufferance of the level of the astrocytes was stronger than at the level of oligodendrocytes. Then we moved to our second work, that was published last year, where we analyzed the data specifically on the activity on astrocytes, and especially we went to investigate GSK3 beta activity. And what we found out that when there was the overexpression of laminin B1, there was an increase of GSK3 beta phosphorylation, so an inactivation of the enzyme, and a reduction of beta-catenin phosphorylated form. But this did not correspond to an increase of its active form, and it could not activate the VNT beta-catenin brain survival pathway. So somehow all the VNT beta-catenin brain survival pathway was downregulated, inactivated, together with the alteration of cyclin D1, so acid cycle progression alteration. So a sufferance that was continuing specifically in the astrocyte cells. Then we did analysis, and PPAR gamma, as shown also in other neurodegenerative disease, in our model of expressed laminin B1 was overexpressed. It was overexpressed, and this paired with an increase of P27 and a block of cell cycle progression with a decrease of cyclin D1. At the same time, still astrocytes overexpressed laminin B1, several models. There was a reduction in Ki67, so also cell proliferation was negatively affected. We also tested in these models the cytotoxicity, and we saw that there was a reduction of viability, an increase of cytotoxicity, and induction of apoptosis in our astrocytes overexpressing laminin B1. So in a way, something that has already been shown in literature, a toxic effect of laminin B1. So a pro-apoptotic also activation of GNK signaling pathway. Together, these arrived with the description of an immunoreactivity showed thanks to GFAP Vmin1 in laminin B1 expressing astrocytes, indicating a reactive phenotype. About the reactive phenotype, we had a very nice talk with Prof. Bujani, and what is interesting is that all the cell models show a reactive phenotype. We are not sure that it's just a normal activation in postmortem brain or a reactive phenotype, but that's what we have been observing in the cells up to now. But again, this was a stress system. And this is still in the first part of the presentation, but this is done on the skin fibroblasts of the patients overexpressing laminin B1. So our patients until now were a duplicated patient overexpressing laminin B1. As you can see here, we have also in the fibroblasts, even if they are not affected specifically in the disease, morphological alteration similar to the one we saw in the astrocytes-like cells overexpressed lamin B1. Of course, if we overexpress even in wild type comparable fibroblasts, we see a very deep alteration similar to the one we saw with the extreme level of lamin B1. Then we were, let's say, lucky enough, the Italian group to enter last year in the biggest neuroscientific Italian network of ever, where we received really big funding, 130,000 million euro for studying neurodegenerative and ischemic disorder related to neurological system called MESIS. And inside this big network done of 500 different researchers in all Italy, we started a collaboration with Prof. Claudio Canale of the University of Genova to analyze even better the quality, the physical property of the cells of our patient with atomic force microscopy. Because something we're trying to do is to understand whether the fact that we're not only facing a leukodystrophy, but a laminopathy. So we have to understand if there are similarities with other disease where the cells are affected specifically due to the alteration of lamin B1 levels. So thanks to the use of the microscope, we were able to start to reduce the poroelastic relaxation and diffusion coefficient. And the first data actually are very complex to be understood, and also they're very preliminary. So I show you just some preliminary data to let you understand how complex still the matter here, because if we compare the nuclear region of ALD to compare to the nuclear region of controls, we can see that there is a higher diffusion coefficient between these two groups. So it seems, but this is really something we have to understand, that even if our cells are tougher, we know, because they have a bigger nuclei, it could be that the nuclei and the membrane is altered, so the diffusion is even increased. So we don't know what is happening specifically, but we know that there is a higher level of sufferance in these are fibroblasts of patients with ADLD. These are some other analyses done in differentiating also the cytoplasm, the nuclear part of the cells. And here it gets even more complicated, even for us, because what we start to see is that only the nuclei region do not show the difference we see in the coefficient of diffusion that we see in the overall cells, meaning that probably also the network between the cytoplasm and the membrane of the nucleus could be irrelevant. And I showed this data just really to show you that there are a lot of analyses we are trying to do, but I have no, let's say, conclusion at all about this data. Just really open, let's say, brainstorming about new data coming from the physical part of the cells. Then we started to investigate the inflammation, because we thought since the beginning that even if this is not the context of multiple sclerosis, there could be really a role of inflammation. And we saw both in the ex vivo models and patient cells, there was an increased level of NFK beta and STAT4. Also, the ROS production was higher in ADLD cells. And so we arrived to the conclusion with these two first papers that astrocytes could be really the first altered kind of cells that could then reduce the support to the oligodendrocytes, then resulting in the disease. We also suggested that inflammation and oxidative stress could play a role. And in the second publication, we investigate deeper the specific alteration that astrocytes might have. So alteration in proliferation, an increase of cytotoxicity, and also activation of reactive phenotype in vitro. Then we moved on to, and we can move on to the second part here, thanks again to Prof. Ramazzotti and Dr. Kufi, who is doing a specific PhD on ADLD, and thanks to the ADLD Center grant and to the Ministry of Health of Italy that is the main sponsor for this part of the study, we moved to primary cells overexpressing lamin B1. So we then started different, as you can see now, studies. And what we start to see here is that, first of all, human astrocytes overexpressing lamin B1, this create increased number of cells with NFT4 and FK beta. So again, astrocyte activation and also cytokines, so inflammation. We also show, you can see here down below, that lamin B1 human astrocytes overexpressing lamin B1 has specific alteration, like the one we showed in the cell models and skin fibroblasts. So we were confirming the data. And this was the data I presented you 2 years ago that we did in the cell models. Now we did it with primary astrocytes, and then we'll do it with iPS cells. That, as you can see here, the co-culture of lamin B1 induced human astrocytes with the precursor of oligodendrocytes is already sufficient not to make produce MBP and to alter the life of these cells. So basically, in these models here, you have wild type human astrocytes and wild type precursor of oligodendrocytes, and you can see that there are the both, even if they are on different levels, and the neurons are these neuromimicking fibers, we have the expression of the myelin basic protein. Also, internal control, we have this. Instead, just overexpression of lamin B1 is sufficient to alter the survival of both astrocytes and oligodendrocytes with the reduction of creation of myelin by the oligodendrocytes, even if still here the oligodendrocytes are healthy. And this is data, we cannot go in detail in this because this is an accepted paper that will be out very soon, but we cannot go into the details, but just to show you that both duplicated and deleted post-mortem tissue analysis of brain of patients show specific alteration only on astrocytes, not present, of course, in the cerebellum of the deleted patients, as also shown before in the MRI part by Rayleigh. And this somehow make us think that it was really possible that we are not facing a classic leukodystrophy, but maybe an astrocytopathy. But of course, we are continuing to investigate. Then we started to investigate, as we were focusing on the glia, the activation of microglia. And you can see here in the last video, the orange dots indicates that when there are human astrocytes with transduced lamin B1, so higher level lamin B1, there's an increase of microglia activity. So we started to put together, let's say, a third player here. So also microglia seems to be altered. Then we arrive to the last part of this presentation before, let's say, the conclusion and future direction. That is the iPS cell models. So I have to say that I think we are very lucky because we built up a very nice network between two main facilities, CBO and University of Trento, and they also just received a grant specifically for creating these kind of cells, always inside this Mnesis very nice Italian network. But also we have a long-lasting collaboration with the Korean Brain Research Institute that is helping us a lot to produce ADLD cerebral organoids. So now I'll show you some of our and network data. So here you can start to see the differentiation of the cells into astrocytes. And what we can see here is that we start to see alteration in our patient. In this case, they are all duplicated patient with some blebing in LD patient cells. So we are confirming in iPS cells, astrocytes that have of patient with ADLD, what we saw in our stress models. And here, what you can see that also the shape of the nuclei are very aberrant and blebbing, as we hypothesized in other models. This is an ongoing study. I don't have I don't have the data for this, but just telling that we're doing the mRNA-seq analysis in the astrocytes derived from our patient. And we're trying to analyze which genes are downregulated and upregulated, but very interestingly, some of the network we analyze seems to be very relevant. So both PI3 kinase mTORC and FK beta. Then we have, this is all unpublished data that I'm sharing with you, the work of Trento, that they did very nice work. And it was very nice because they started to work also on deleted astrocytes cells from ADLD patients, not only duplicated. Here, the data, of course, we cannot do statistics. We have very few patients. But what you can see is that it seems from this data that the deleted patient have higher reactive phenotype than the duplicated. But this is really, again, just the beginning of this analysis. And also you can see here specific alteration, stripped, crumbled nuclei that are altered in both deleted and duplicated. Of course, patients show heterogeneity of clinics, and also their cells are very heterogeneous. So it's never easy, this part. But together, it's very interesting for us that different groups with different even techniques, they are starting to show similar things of the sufferance of astrocytes. Then the question of Trento was, is it possible to produce patient-specific regional pathological changes in central nervous system organoids? So they started to create from non-canonical deletion and canonical duplication, both for brain and cerebellum organoids, that they are starting to develop and they are growing very well. So I hope in the next months we'll have some nice data from these parts, and I really thank them for their help and effort in this project. And then we have another part done, all this is done by unduplicated patient. And this work was done by Dr. Shin and Prof. Kosodo of the KBRI. And we have a very nice collaboration. And Dr. Kofi is going there for 6 months to work with them and continue this part, started from our patient cells. And what's interesting is that at the beginning, so the early stages of the creation of the organoids, there was no difference between controls and ADLD patients' organoids. Then we start to have an increased level of lamin B1 in the patient cells, so the organoids of the patients. And also this group started to observe an alteration on ADLD organoids on the astrocytes. But what was very interesting for us, and they confirm, I think, the possibility really that is even possible to imagine, a communication with the oligodendrocytes, that instead in oligodendrocytes we have a reduced number of mature oligodendrocytes in ADLD organoids. So again, it seems that the sufferance of astrocytes reduced the maturation, and we saw the same data on our mimicking neural fibers with primary astrocytes overexpressed lamin B1. So all this data, when it will be confirmed, I think will be very interesting for the next steps in understanding the disease. Then arriving, let's say, to an end, close to the end here, this was the project done with ADLD Center. And the main goal, of course, that was also on the comic I show you in the second slide, is, of course, to find a cure for a patient. We need to have reliable models. You can have the mice, you can have the cells. All the models should be tested, of course, in order to test a reliable drug. And that's where Elisa Giorgio comes in with a very nice project here that was founded by the Ministry of Health, where we are going to test on the different models we are developing drug repositioning strategies, so already available drugs to test if some of these might have an effect on the developing of the disease. The collaboration here, I want to remind, is done also with Prof. Cortelli and Ariacani of the University of Bologna and of ESNB, that is the Neurological Science Hospital of Bologna, and with Gaslini Hospital of Genova. And this was the paper that was just accepted on the 5th of July. That was very important for us. We cannot go deep into this because the editor didn't allow us to show all the details of the paper, but it will be open access and out soon. But after analysis of more than 20 families, with different phenotype of lamin B1, we showed that basically it's not sufficient to have a duplication of lamin B1 to have a per se diagnosis of ADLD. That means that there are patients with a duplication, but with an unhealthy phenotype. So they have a duplication, but in the brain, the duplication is silenced so that there is no overexpression of the protein. So this could affect very, very much with the genetic counseling. And I think that we'll have to really focus on the fact that is the overexpression, not the duplication, probably the thing we have to look closer to. These are instead just some ideas we had because of the level of lamin B1 and also our background in cancer, not only in neurodegenerative disorders. We started to investigate and compare where different situations of alteration of lamin B1 and lamin B2 were overexpressed or decreased in different cancers or other laminopathies in order to really understand the mechanism. A very nice work just published was instead the role of lamin B1 as a key modulator in the developing and aging brain, work done in between our group and the Korean Brain Research Institute, again to understand not only the level of lamin B1 in pathological situation, but in developing and in developing brain in aging brain. This slide I put here just to thank Margot for her effort for this year. We were very glad to host the 2022 First International Symposium. It was very nice. And even if we are online, it's very nice. And it has been also very nice until now. Thank you. And after this meeting, I think we did a nice collaborative effort. So we had a collaborative review led by my PhD student Irene Neri, where we discussed all the models used until now, until that day, now it's already old, about ADLD. So I think that the collaboration is really the key of success. In 2023, I have this occasion also to talk 1 second about our ADLD Italian Patient Association that was created many years ago, but he had the first big meeting in 2023. Here in the QR code, you have the link to all our research and also to the association, hoping that we'll find a way, even if, of course, there is the gap of the language. And we discussed this also with Bish and other people involved in ADLD Center with your community. And now I just want to conclude. There are many names here because we are a big group. ADLD is one of our focus, but as the long list that Margot read you at the beginning, we do many activity at the Anatomy Center of the University of Bologna. But in the picture, you show the people that are specifically involved in ADLD, Prof. Ramazzotti, Dr. Mongiorgi, Rusciano, and Kofi. They were very important in development of all these projects, and also Prof. Cocco and Prof. Manzoli. But even more important, I would say the blue column here, all the collaborators around the world from different places in Italy, but also Bujani in Amsterdam, KBRI, Cibio, Giovanna Vaula in Torino, and Gaslini Hospital, and of course Elisa Giorgio and Maria Lodorzonna in the University of Pavia. Here, down here, all our grants, ADLD Center, the Ministry of Health, Italian Ministry of Health for the grant to Elisa Giorgio and me for developing other drugs, and the Mnesis network. So that's all. Here you have my contact, and I'm ready now to take any questions. Thank you.
Thank you, Stefano, for sharing all that work from your group and your colleagues. I can open the floor to any questions in the room. Pastor?
Stefano, how are you?
Good. Hi.
Nice to see you with longer hair and with a mustache. I don't think you had that 2 years ago.
I don't remember, but thanks.
So again, I don't know. So I think, what you said is right. If you look at autopsy patients, the ADLD samples, they're characterized by the astrocytes, which look very abnormal, right? The processes are blunted, they look plumper. So in your cell models, did you look at the overall morphology of the astrocytes? How do they look? Do they look abnormal or they look similar? I know you show the nuclear morphology, but what about the overall cellular morphology?
Yeah, that's a very good question. Actually, all the morphology is altered, and it changes, I would say, together with the level of overexpression on lamin B1. So when we selected only the cells overexpressed lamin B1, of course, the nucleus is very altered, but basically all the body is shrinks and it becomes with less capacity of having the specific shape of an astrocyte, and the cells tend to die and to reduce their activities. So all the cytoplasm is altered. Yes.
Okay, great. Thanks.
Thank you, Pat.
So just following up on that, if there's significant astrocytic dysfunction, why are we not seeing a vascular leakage? So other disorders of astrocytic dysfunction like GFAP, neuromyelitis optica, they all tend to have vascular leakage, which is macroscopic, and you can see it on MRI with post-gadolinium enhancement. Why don't we see that with this disorder?
Thank you. I didn't get your name, but thank you for the question. So I think this is a very good point, and I think that the problem of the leaking is also related to the time of when there are the observation with the MRI. I'm not the clinician of the patient, so I'm not responsible for the MRI. So what I can tell you that we didn't develop the vascular model yet, so this thing in vitro is not observable yet. I can also tell you that in a post-mortem tissue, there's not specific alteration, but it's also there are even forms where just the astrocytes are altered, and as it can be a late onset disease developing very in a long time, our theory or possibility is that the alteration of vascular parts is not that dramatic, so it's not seenable. But this thing, I think, should be analyzed following youngest patients, let's say, we have in time to be sure of this.
Hey, Stefano, we're just checking if there were any online questions. But I have a question about your models and some of the signaling work that you looked at in the cell models with kind of the drastic overexpression. Have you been able to recapitulate those in the human models and the organoids?
Very good question. The answer is not yet, because we have two things. Still, the models need to be matured to test the signaling part completely. We started and we see similar things, but we need to continue that. And second answer to your question is the RNA-seq data. We're developing the data, and once we'll have that data from RNA-seq of astrocytes, and we'll know which genes and which signaling pathways are altered, downregulated and upregulated, I think then we'll have a specific answer to your question. That is also our question. Thank you.
I'm really interested to see. Yeah, I'm really interested to see what as you kind of finish building out those models and see what you find, what that means for drug repurposing and pathways. Because I think we're going to find a lot of answers there.
I think that really the challenge is really to develop reliable models, and at the same time to show in all the different models, get them as similar as possible to the patient and find out the best druggable part. So yeah, I agree.